My name is Grace Erickson, and I'm presenting on glucose uptake in myocytes or muscle cells following an environmental contaminant exposure of DDT. And this just gives a little bit more context and is a little bit longer than my original presentation title into the actual data and the experiment. So diphenyl trichloroethane, or DDT, is an insecticide, and it was once used on crops, but then it was later used to mitigate vector-borne diseases such as malaria. And DDT was manufactured in plants across the country and the world. And it is an organic pollutant, which means that it does not degrade quickly in the environment or in tissues. And it is not acutely toxic to mammals. So that's why it was very popular. But it was later classed as an organic pollutant for doing widespread ecological damage and possible health effects on mammals and other organisms. So, as I previously stated, DDT has a long residence life within the environment, and it bioaccumulates in both the environment and organisms. And the diagram shown in the bottom right corner shows the process of bioaccumulation and how that might happen within an ecosystem. And humans, especially, or other organisms, are exposed through contact with contaminated soil, consumption of contaminated fish, or consumption of contaminated vegetables, as well as a few other minor exposure routes. And this is important and kind of ties in because studies have shown DDT in fish populations throughout water systems across the world. So why is this important to us in the Gratiot County community and area? Well, the old vesicle site in St. Louis, Michigan is still a site for bioaccumulation. And individuals living within this area are at a higher risk of DDT contamination. This image is just of the old vesicle site in St. Louis on the Pine River. So DDT and DDE, a metabolite of DDT, may cause cancers, endocrine dysfunctions, reproductive health issues, and metabolic disorders such as diabetes. This is again important for us in the Gratiot County area because mortality from diabetes is higher than the national average. It is 5.6 times higher for females, and it is at average for males. So what is diabetes? Diabetes is a disease involving controlling the amount of glucose levels within the blood, and that the blood glucose levels are too high. And insulin is the main hormone for regulating the amount of glucose within the blood and making sure that the glucose in the blood is low. There are three main tissues for glucose uptake and storage. That is the liver, adipose or fat tissue, and skeletal muscle. And there are two different types of diabetes. There's type 1 diabetes, or more commonly known as childhood diabetes. And this involves the amount of insulin secreted into the blood. But type 2 diabetes is more commonly known as lifestyle diabetes, and this involves insulin resistance within the body. So why study skeletal muscle? Well, in the postprandial state, or right after consuming a meal, skeletal muscle clears about 80% of glucose from the blood. And also studies have not yet shown the effect of DDT on skeletal muscle. The overall goal is to see changes in glucose uptake from muscle cells that were dosed with various concentrations of DDT, with the prediction being that there will be a negative impact on glucose uptake. To test our hypothesis, we used L6 skeletal myoblasts, which is an immortalized form of rat cells. And the cells were maintained in DMEM, which is just a media to make sure that the cells have all the nutrients that they need to survive. And cells were kept in an incubator at 5% CO2 and 37 degrees Celsius, mimicking body conditions. And the image just shows skeletal muscle cells plated on a plate in the lab. So the cells were plated in a 96 well opaque plate at 0.5 times 10 to the 5th cells per well, and an image of the 96 well plate is shown in the bottom right hand corner. Solutions of DDT and DMSO were made, and then cells were given 5 days to differentiate, and the cells must be fully grown in order to be able to metabolize glucose, so that's why they are given 5 days to differentiate. And treatments were added 48 hours prior to the experiment. Then, in order to measure glucose, the Promega glucose uptake glow assay kit was used, which uses a luminescent form of glucose to measure how much glucose is taken up. And an image of the protocol can be seen on the right-hand side of the screen, where we use a luciferin detection solution, and luciferin is what is going to be luminescent, and we add other ingredients to it, and then add this solution to our samples of cells, and then we allow it to incubate for 60 minutes in order for glucose to be uptaken within the cell and metabolized, and then we read the results. We measure glucose 6-phosphate. So in the reaction seen below, we have glucose being transformed by hexokinase into glucose 6-phosphate when glucose enters the cell. This allows glucose to be trapped within the cell and to create a concentration gradient to keep bringing glucose into the cell. So that is how we know that we are measuring glucose within the cell and not just glucose added to samples. Then total luminescence was read by the SpectraMax i3 plate reader. So as can be seen in this figure, the control group in DDT1, which represents 100 micrograms per ml of DMSO, DDT2, which represents 50 micrograms per ml of DDT, and DDT3, which represents 100 micrograms per ml of DDT, were all compared to luminescence in relative light units. And bars represent standard error, and the p-value is 0.74, showing no significance. The more luminescence indicates a higher amount of glucose uptake. And a negative control study was run to determine that insulin did indeed have an impact on the cells. Overall, there was no difference seen amongst the control and test groups, meaning that DDT had no effect on skeletal muscle glucose uptake. This could be for a variety of different reasons, one of them being the sensitivity of the assay. The standard gold method of this study would be to use radioactive techniques, which was not available to us. And also, the previous study completed with this technique showed a 2-fold change in glucose uptake. And so if we were measuring anything or saw anything below this, that would mean that it probably wouldn't have been able to be detected. Also, the sensitivity of the cells could be a problem for this. Typically, C2, C12 cells, which is another immortalized cell line, would be used. But in the process of immortalization, they lose all sensitivity to insulin. So instead we choose L6 cells, but these still might not be as sensitive to insulin as live tissue or tissue samples would be. Or it could be the possibility that DDT does not have an impact on skeletal muscle glucose uptake, and instead it is working through a different pathway to impact diabetes. Possibly, the cells that we are measuring currently may take up glucose, but possibly over time they may not metabolize glucose as fast. Our study was over a 48-hour period, so if we lengthen this time, we might see more of the impact on metabolism. Also, other glucose uptake and storage tissues might be involved in this, such as adipose tissue or fat tissue that stores glucose. Or the liver could be impacted, which has a variety of different reactions involving glucose metabolism, such as gluconeogenesis, glycogenolysis, and other metabolism reactions. Or DDT could be impacting type 1 diabetes and working through the pancreas and decreasing the amount of insulin secreted into the blood. Possibly, future directions of this could be to measure hexokinase. And as previously shown, glucose is transformed into glucose 6-phosphate by hexokinase when glucose is entering the cell to trap glucose into the cell, and also to create the concentration gradient to keep glucose being taken up within the cell. And so this would be an indirect measurement. In theory, higher levels of hexokinase would mean higher levels of glucose uptake. Or other tissues could be studied as well. Adipose tissue or liver tissue or pancreas could be tested, or we could lengthen the time of our study to determine more long-term effects of this. I'd like to thank Dr. Ball and Dr. Harfman for picking up this project and seeing this project through. And I would also like to thank Dr. Harwood for lending us her DDT samples and making this possible, and the IPHS department for any financial support and other support in creating this project, and Alma College for allowing us to present our research and for giving us the opportunity to complete research. Any questions?